Are you seeing the presenter mode now?
It is not in the presenter mode.
Now
Yes,
it is.
now it's it. Yes.
Okay. So I will start because the audience includes students and those who are not working in malaria. So I will start a brief introduction. Like it, the talk will be like less focused on my research and more on the background, like what is going on in malaria field, how the vaccine works, on these kind of things. So please feel free to ask any question if you have any problem. I'm happy to take questions as they arise. So the title of my talk is Malaria Vaccine and Approach Matters. And you will see through my topic, through my presentation, that how we approach malaria as a problem and how we design vaccine. And the vaccination approach is important. Why it is important? I will go through this step by step so that let me know like if you have any problem anywhere understanding that. So to start, these are my talking points. First, I will start by giving you an introduction about vaccine, and then introduction about malaria and malaria vaccine. Then I will touch on a little bit about the research that I am doing on malaria vaccine. So just to give you a brief history of the vaccine, so I will take you back when the work started and how the word vaccine was it is developed. So, as most of you might be knowing, that Edward Jenner is known as the father of vaccine because here what you are seeing is uh, Edward Jenner is immunizing a 8-year-old boy with the cowpox, but he didn't. The name vaccine didn't start at that time until it was Louis Pasteur came about 100 years after that, close to 100 years after that, and he developed the rabies vaccine, and he named in honor of Edward Jenner this process of immunization as a vaccination. That's how the vaccine started. But this is, if you see this process, this is the word vaccination. It comes by Westerners like in 1800 early 90s. But what happens is that this process was age old. So I will take you back close to like 2,200 years, with like 200 BC. And during this, like there are records that says that like people have been using this strategy to vaccinate people, but without knowing that this is vaccination. So this is one historical record in how people used to do in China. They were taking the pustules from people who were having smallpox. They will scratch it and they will scratch on other people like healthy people. That's how they used to vaccinate them. And this is another example where what they were doing is the person who is putting in the nose the dried pustules from a smallpox patient into the nose of a child so that they will develop immunity against smallpox. So this was the practice like almost 2,200 years ago. So the people knew that that's how people get vaccinated. So now I don't want to focus that why vaccine is important. After this pandemic, everybody is aware why the vaccine is important. And I will just end this slide with this quote that prevention is better than cure. So if you prepare yourself and you can prevent getting infected and getting the disease, it is better. So and everybody will agree with that. So how the process of vaccine works. So in these slides, this is just a kind of textbook picture. They describe in the left side, you are seeing a person is unvaccinated. And then what happens is if a person is unvaccinated and he has exposure to pathogen, his immune cells like your B cell and T cell, these are the basically the immune cells that I'm just taking a subset. I'm not going into the detail, just giving you a elementary view how the immune system works. So this B cell and T cell, they respond by generating memory and by producing antibodies. But by the time they respond to this pathogen, the person is already sick. On the other hand, how the vaccine works. So if a healthy individual is vaccinated with part of the pathogen or like the attenuated or killed pathogen. so that the immune system now knows what it's looking. So what they will do is they are already primed. So next time when the individual encounters a pathogen, the cells are already prepared. The immune cells are prepared, and they know which things to attack, which pathogen or microbe to attack. So they start acting on it, and so the person who is vaccinated, they successfully eliminate the pathogen before it creates the disease. So that's the benefit. So here I will quote from Louis Simpson that the aim of military training is not just to prepare men for battle, but to make them long for it. So basically, what we are doing by vaccinating is we are training them to search these pathogens and kill them before they can create any problem. So that's how it is important, and that's how it works. So now, after you know how vaccine works, how vaccine started, and now let's see what are the types of vaccine that we have so far. The first one is the live or attenuated vaccine. This is a kind of like classical way of that people are vaccinating people, where they uh, basically they use either live or attenuated means they inactivate it either by heat killed or by chemical treatment, or any way they attenuate the parasite so that it can infect, but it cannot cause any disease. But there are some, like this vaccine is highly effective and it requires a cold chain, which is one of the drawback for this kind of vaccine. So because in remote places where there is no electricity and like problem with the transportation, the cold chain creates a kind of problem. Okay, but this is more effective vaccine, and some examples are the smallpox, chickenpox vaccine, and there is another is killed or inactivated, and this basically this killed or inactivated, it will give you an exposure of the pathogen, but you need boosting because the pathogen will not develop and it cannot express all its antigen. So some of the example of this are like hepatitis A, flu vaccine or polio vaccine, and rabies vaccine. There is another vaccine is the subunit vaccine. In subunit vaccine, what happens is you take part of piece of the pathogen, either it's protein or it's polysaccharide or something that is like mostly it is protein on surface of the pathogen, and that is important for the microbe of the pathogen. So what you do is you take that and you train your immune system. You inject to the individual and they develop antibody or activate T cells, and that's how you train your immune system. And some of the example of this is hepatitis B and whooping cough vaccine. And these are comes under subunit vaccine. And then another vaccine is the toxoid, and this is most common example is the tetanus vaccine, where you take the like attenuated form of the toxin produced by the pathogen and you use that so that that cannot cause any once like you have immunity to that, it cannot cause any kind, the toxin will not have any effect. So toxoid is there is another way, another form of vaccine. Then there is peptide vaccine, where people take basically the part of the protein. Those are highly immunogenic and they are unique to the pathogen, so that they can, if, once you generate antibody response or T cell response against them, you can attenuate the pathogen. So some of the example of this are malaria, HIV, and influenza. I will discuss some of them in when I discuss the malaria vaccine. But this peptide vaccine, it requires like a strong adjuvant because they are small peptide, like 7 to 8 or 9 amino acids. So they themselves are not very highly immunogenic. So we need to adjuvant them. They provide something that is going to boost their antigenic property. So you need peptide vaccine. You need this kind of. Those are the drawbacks for peptide vaccine. Then comes the recombinant DNA vaccine, which basically gives you advantage that you can stitch multiple antigen and make a long like DNA vaccine that can express multiple antigen. But like clinically, it is not that very successful, but experimentally, this is very highly successful. And some of the example are HIV and Ebola, or like your respiratory virus like H1N1. This vaccine, they use this technology. Then the other is like naked DNA technology, where they basically mass produce this DNA particular gene that's going to be immunogenic, and that's they inject this DNA, and the DNA get expressed in the host and produce the protein, and the host mount immune response against them. And then currently, as all of you might be knowing, because of the SARS-CoV-2, RNA has been used for the first time in human. It has been used before in animal models, but not in human. But for the first time, it has been most successful in human as a vaccine. So RNA has been used where it is just the messenger, so that it don't get integrated into the genome, but it will produce the protein that is required to generate the antibody or immune response against. So these are the different kind of vaccine. So this slide basically describe how the vaccination process has been evolved. Initially, in the beginning, it is just like the empirical approach where you take the like dried or attenuated or maybe some non-similar but non-related pathogen and immunize them. Like if you look at the like smallpox, people were being immunized using cowpox, so they are protected against smallpox. These are related virus, but they infect different individual. So that's how they were doing. But there is these are just empirical approach. There is no scientific basis at that time when they started this process. So then it evolved. The process of vaccination evolved once it was discovered as a vaccine, and they knew that there are advantage of vaccine. And then with the understanding of molecular biology after the when they decipher the nucleic acid DNA sequence, they knew your DNA from DNA to become RNA, RNA to protein. All this like central dogma, all this understanding aided the process evolved in the evolution of the vaccine, then started recombinant DNA. And then when people like understood what are the different parts of the pathogen, they're important and they can be utilized as a vaccine, that's how different kind of vaccination approach started, like glycoconjugates, reverse vaccinology, and currently the next generation technology, which is basically your synthetic biology, DNA and RNA, basically stuff. And now there are also like structure derived vaccine where they generate immune response against structural features of a protein, not only the naked protein or the amino acid sequence. So these are the next generation technology. They are still like some of them are being used, but most of them are in experimental mode at the moment. So as I gave you a brief background on vaccine, how it evolved, what are the different kind of vaccine. Now let's talk a little bit about malaria, because that's the topic that I'm going to focus more on. And just to give you an idea why malaria is important, this is a map that shows the like cases of malaria, and I just summarize this in 3 sentences. The first one is like this is estimated like 228 million cases happen malaria globally, and almost like 500,000 people die of malaria each year. They are mostly them. They are like child, like less than 5 year old uh, age. And the third point that I wanted to focus is because this presentation that I'm giving, and I am from Odisha. So Odisha accounts for more than 80% of malaria cases in India. So that's the endemic state, most endemic state in India for malaria. And this is what another point that I want to highlight is half of the world population are so at some level of risk of getting malaria. So that's close to 3.5 billion people. So that's a large number. So it is a significant problem. Right now, people are focused on COVID as it is like it's a pandemic, but definitely malaria is not less than a pandemic in terms of its global mortality and morbidity. So another ongoing problem with the malaria is that malaria is normally spread by insect vector that everybody knows. A mosquito are responsible for spreading malaria, and these mosquito, they are getting resistant to all the insecticides. Here I am giving the most like the 4 classes of insecticides that are being commonly used: the pyrethroids, DDT, and carbamates, and organophosphate. So these 4 classes of insecticides, and what you are seeing in the map, which are in red, in those areas, including India, you will see that all the mosquito are resistant to all the 4 classes of insecticides. So This, when you use these insecticides, the mosquito are not going to die. They are going to spread the disease. So it is another big problem in the control of malaria or elimination of malaria. So in this scenario, the best weapon we should have is the vaccine that's going to be effective, so that even though mosquito are transmitting the disease, we should be protected if we are immunized. And this slide is just to give you an introductory idea about the life cycle of the plasmodium, how it happens. And as you know that plasmodium has two life cycles. One, it happens in the mammalian host, and as we are human, are also host. And the second part, the sexual cycle, happens in the mosquito. So it starts with the bite of an infected mosquito, and this infected female mosquito, when it seeks blood meal, uh, it it inadvertently injects sporozoites, the form that is insect form of the malaria parasite. Then these sporozoites migrate to the liver, and in the liver it transform, and one sporozoite produce close to like 30,000 merozoites. And these 30,000 merozoites, they come to blood and they invade your RBC, and then they cycle. Then they develop into skyzones, and one infected RBC produce like close to like 14 to like 16 merozoites. And this cycle goes on. And during this cycle, we normally get, when the get these symptoms of fever, chill, and this cycle is periodic. So that's why, depending on the species of the malaria, we get like periodic fever, like every 48 hours or every 72 hours, depending on which species of malaria we are infected with. And some of these blood form, they differentiate into gametocytes. And then when another like female mosquito takes a blood meal, they take these gametocytes along with the blood meal, and the sexual cycle starts again in the mosquito. So, like, as you can see this cycle, there are certain bottlenecks in the parasite's number. So I will describe that bottleneck here. So on the y-axis, what you are seeing is the number, the like average number of parasites. So I will start with this, that from the liver it is coming to the blood, and the blood, like from the liver, the number is basically in like hundreds or maybe tens, between tens and hundreds. But in the representation is so high that from the liver, once it comes to the blood, in the blood, the number of the parasite can goes to trillion. And then, like when it is taken up by the mosquito, the number can goes to less than 10, like less than like close to five oocyst. Like, so this is another bottleneck for the parasites. And this was one of the bottleneck for the parasites. And then this liver form, when they release in the beginning, like, no, so sorry, this oocyst in the mosquito, they produce sporozoites, and these sporozoites, there are only few sporozoites, like 10 to 100. They get injected into the mammalian host. So this is another bottleneck. So these 3 points are the bottleneck where the parasite number is very few. So we cannot directly control in the mosquito. There are some approaches people are using that I'm not going to talk about that in this seminar because of the time limit. But I'll focus on like uh, particularly this stage, and how we can study this bottleneck, and how we can control the parasite before it comes develop in the liver, and before it gives to the these trillions of blood infecting parasites. So this is just to highlight the number, the difference in the number from tens to trillion, to again like less than 10 to close to like 10 to 100, and then it continues. Okay. So what it means, if you study this bottleneck, what it means for the vaccination approach. Another thing that I want to highlight in terms, apart from these numbers, one thing that you will notice, that the parasite is traveling from liver to blood, and it is changing its different form, shapes, and morphology. And also, like, it is so adapted, this parasite, is that it produces different kind of different number of proteins. So it makes it much more complicated when you think in terms of vaccine. I'll come to that in the next slide. So what are the complexity you should consider while designing a vaccine? So I'll give you an example. This is, everybody can recognize this picture because this is now in the fashion, that this is the SARS-CoV-2. and it has only 28 genes. Let's take a little more complex organism, that is an E. coli, and it is close to 4,000 genes. Okay, so and then let's talk about the malaria parasites. And here I have represented three different forms. One is the ookinete, it normally happens in the mosquito. And then this is the sporozoites, it happens in the mosquito, but it interfaces with the mammalian host because this is the form that infect human. Okay, and then this is the merozoites, that's the blood infecting form. You can see the difference in their shape changes, the morphology changes. And this malaria parasite has more than 15,000 genes. So when you design a vaccine, how you decide that which gene you are going to take, how you are going to immunize, then how is going to target. Say you design a gene that is important for this stage or maybe this stage. So if one parasite escape this stage, then your vaccine is ineffective, right? So these are the complexity that you need to be thinking about when you design a vaccine and when you design a vaccination approach. I'll give you an example. So for the malaria vaccine particularly, RTSS, this is the one vaccine that has been approved by FDA, and it is now being rolled out in Africa. And just recently, it has been passed that it can be given to kids like globally, and it has an efficacy of 33%, which normally went down in 9 month time period. So it can be important. So think about it. If malaria is seasonal, right, like in Africa, it is seasonal. Like it happens like rainy season. Most of the malaria cases happens in rainy season, and it die down the dry season. But look at in the scenario in India. In India, it happens throughout the year. There is no seasonal pattern to malaria. So if you design a vaccine that is effective in, say, in Africa, it's not going to work in India because it's not seasonal. So in Africa, if 33% efficacy vaccine you are giving in high peak season, then it can reduce the level of the case number, because in the dry season there will not be any malaria cases, any transmission. But it will not work in India. Then recently there is another vaccine that is also a kind of sovereign vaccine called R21 matrix M, which is 77% efficacy, and the protection lasts close to like 1 year. But this is in phase 2 clinical trial, and they are trying for phase 3 clinical trial. We don't know what will happen after phase 3. Most of the like vaccine, they show in the beginning, beginning they show a lot of promise, but when in large scale study, because of the diversity, that goes down, the efficacy goes down. So you have to see, wait and see how it is going to be. But it is really optimistic that 77% efficacy is really good protection, good efficiency, particularly for an eukaryotic vaccine like malaria. Okay, and I want to highlight that these both vaccines are subunit vaccine. So think about that. They're taking a protein or a part of a protein to make the vaccine. And imagine that this parasite keeps changing this protein. Experiment will have the protein as well as like its shapes, and the duration of their development is different. So can we use whole organism as a vaccine, like a classical approach? Where people use the whole attenuated form or whole organism. So can we use that? So that's the question. And then I'll come to that. And this is highlighting some of the, like, this is a lot of information. I don't want you to focus on all the information. Just what you want to focus is all these boxes. Most of them are subunit vaccine. You can see the red boxes, red squares. And these are like targeting different stages of the parasite, because the parasite has different stages. The pre-erythrocytic stage, basically the liver stage, when the parasite is silently developing in the liver. And the most successful vaccine so far is this RTSS that I told you. That is based on the CSP. That's a protein that is covering the sporozoid surface. And this vaccine has 33% efficacy and is now clinically approved. And then there is asexual blood stage, where the parasite produce trillions of blood infecting parasites. So most common vaccine is against this stage MSP1, which is normally a protein called merozoid surface protein, and these numbers are basically the amino acid sequence they are taking from that protein, and they use, but these are not that very effective in particularly human, though they are effective in rodent model or experimental animal model. They are not very effective in human trials. So they are still going on. The clinical trial is still going on because something is better than nothing. So we need to have as many kind of vaccine available, which will aid into your little more on whatever vaccine existing. And there is another one called transmission blocking vaccine. So this is a kind of Trojan horse kind of the process where you develop. You normally don't encounter the protein that is being expressed in the mosquito in your body, but you are immunized against those proteins. Like here it's given the like name PFS 25 or PFS 45 48. These are the protein. They normally get expressed in the mosquito stage, but people are like immunized against this protein so that they will have antibody. So once the parasite takes the blood meal, it will take the antibody along with that, and the antibody is going to kill the parasite in the mosquito stage so that they cannot transmit the disease to next person. So these are the different strategy that people are using to fight malaria. So I will just focus from here onward. I will focus on how different approaches of whole sporozoid vaccine. That's the whole range of vaccine, and what we have learned so far using these approaches. So I will first describe one vaccination approach called RAS, or radiation attended sporozoites. This vaccination approach started in 1967. So what we do normally is we immunize uh, humans like with infected mosquito, and these infected mosquito are radiated with X-ray or gamma ray. So what happens is when you radiate this mosquito, infected mosquito, the radiation is so damaging that it breaks the DNA of the parasites. Okay, so these parasites, then you use this mosquito, and this mosquito, uh, people are being fed by this infected mosquito, and this parasite will go to the liver because they have all the necessary machinery. But this, after initial development, they will stop there. Okay, so in this approach, you need normally 1,000 mosquito bite to, and once you get that mosquito, 1,000 number of mosquito bite, you get a protection that lasts up to 10 months, only 10 months. Okay, so you can imagine how like practically impossible it is to immunize a lot of people, where you need to infect each and every individual with 10,000 mosquito, and the protection will last only for 10 months. Okay, then another approach started after the generation of lot of tools like molecular tools to transform the parasite, and the whole genome sequence of the plasmodium was known. Then it started developing another like generation of vaccine called GAPS, or genetically attenuated parasites. So once the molecular tools were developed, so and this concept was started in 2005. So what this concept uses is like they target specific genes which are important at particular stage in the liver development. So you knock out that gene, ideally, conceptually actually, the parasite should stop at particular stage, like early liver stage, mid liver stage, or late liver stage. Because imagine when the parasite is developing in the liver, is going through this first trophozoites, then like skyzones, then it's like it release the merozoites, the blood infecting form. So they express a whole repertoire of antigens. These are unique to these different stages. So if you have immune response in all these different antigens, then you are most strongly immunized. Okay, but the problem with this approach is that nothing is 100%. In biology, nothing works 100%. Like you knock out the gene, it doesn't mean that the parasite will be completely attenuated. Some of the parasite definitely can make into the blood and start infecting. So this, in terms of when you think about a vaccine, you created a genetically mutant parasite and hoping that it's not going to go to the blood, but it doesn't happen. And if you are immunizing a group of population people with that parasite and they get infected with the blood, then it is a kind of problem for particularly for a kind of vaccine. People will not go for that. They will basically is they knowingly they're getting infected. Okay, then the third one, it's called CVAC approach, or chemoprophylaxis vaccination. This was started in 1977, but it picked up in 2004. So what happens in this is people are get infected with wild type parasite, it develops normally during the liver, but they get treatment with the drug. They get prophylaxis of the drug that's going to kill the parasite, particularly when it is developing in the blood stage. So it will kill the parasites. So they will have exposure to antigens against all this sporozoite as well as all the liver stage and some of the blood stage antigen. So in this approach, what we have seen so far is it requires only 60 mosquito bite, not unlike the RAS, it is 1,000 mosquito bite. It requires only 60 mosquito bite, and the protection lasts for more than 2 years, which is highly encouraging. But still, the logistical challenge remains that you have to infect people with mosquito bites, and transporting infected mosquito for immunization is a big logistical problem. So you need to generate a different kind of approach. So with that background, I'm coming to the research that I have been doing for last 6 years, something like that. That I will tell you what we are doing and how we are trying to improve this approach, this vaccine. So I'll just quickly. I'm not going to all the background because there are a lot of data that I don't. Each of data description of each of data will take a lot of time. So I'll just quickly tell you some of the story that we are working normally on the biology of the parasites, how the parasite develops in the liver. So, so basically what we are doing is we were studying one gene called ATG8, which is a kind of autophagy related gene. And I'm just giving you an idea that this gene is highly conserved in all different species, Plasmodium falciparum, Plasmodium vivax, Plasmodium nolgei, Plasmodium eoli, Plasmodium berghei, and Plasmodium savaudi. These are like, these bottom three are the rodent parasite and the top three are human parasites. And it's highly conserved. And we were creating a mutant for this gene to study its function. And we observed that when we created a mutant, the wild type develops normally in the liver, but the mutant develops very poorly. Okay, but when we infected this with this sporozoids basically the forms from the insect that goes to the mammalian host, we observed that the mice which receive the wild type of the parental parasite, they all get infected by day 3. But the mice those received the mutant, they like develop bloodstream infection like starting from day 7, and some of the mice they didn't develop blood infection at all, and it took on an average 12 days to develop the patent bloodstream infection. So this brings to our attention that this comes to our attention that this parasite stays for a longer period in the liver. Like this normally, I want to highlight here that the rodent parasite normally this development from entering the liver stage to coming to getting out of the liver and infecting blood, it takes 2 days. In human, it takes 7 days in Plasmodium falciparum, but in Plasmodium vivax, it takes 7 days. Sometime the parasite stays in the liver in the form of hypnozoites and comes up after years, maybe months. So that's a different time frame. But on an average, in human it is 7 days, in the rodent it is 2 days. So in our case, we created a mutant in the rodent parasite. They stays more than 12 days, like 7 to 12 days. So that gives us some idea that if it is staying longer, it should be better because the immune system in the liver is getting exposed to these antigens for a longer period of time, so they can mount a better immune response. So we wanted to study that because this is now an interesting tools to study. So we did an experiment. What we did is we combined this genetically attended parasite with chemoprophylaxis. So we immunized them on day 0 and after 10 days with 2 doses. And after each immunization, we treat them with the chloroquine. And then on day 21, post first immunization, we challenge them with the wild type parasite. And here we use the different immunization dose. We immunize them with tailvein IV injection of 20,000 dissected sporozoites and challenge them with 10,000 wild type sporozoites. And and so we did here, and then we look at blood smear and look at positive and negative. If the blood smear is positive, they are not protected. If the blood smear is negative, they are protected. Similarly, we challenge another group of mice on day 80, which is basically memory challenge, and another group we did on after 6 months to look at how long the protection lasts. in this model. And we published this thing already recently in NPJ Vaccine journal. So I'm showing you the result here. What you are seeing here is the black one is the wild infection control. These are non-immunized, but they were infected with the wild type. So they all came to the blood by day 3. But as you can see here, the wild type CVEC, the basically the this is the mice, they received the wild type under the drug cover, and the green, the mice, they received the ATG8 mutant and under the drug cover, and the blue is the wild type, they didn't receive any immunization but only drug. Okay, this is the control. So as you can see, the infection control and the drug control, they all came up by day 3 and day 4, but these two, they're completely protected. Okay, so this is the effector response, like day 21. But when we looked at the memory response, day 20, day 80, or you can see day 80, we saw that the wild type lose, like 40% of the mice lose protection in the wild type, but 100% of mice, they were protected with the ATG8. So our hypothesis is that when they stay in the liver for a longer period of time, basically they are protecting better. So that's working. So, but we don't know what is the mechanism here. And this is normally we look at positive or negative in the blood smear, but to be sure, you have to validate them using molecular tools. So what we did was we validated by doing PCR on the blood of these mice, and we saw that there are 5 mice in the group, and as you can see in the wild type CVEC, 2 were positive. That's the 40% positive, and then in the ATG8, all of them were negative. And we took mouse beta actin as a control gene here, just to see our PCR is working. And in addition to this, we tested again. We took the blood from these mice and transfer, because if there is a subpattern level of parasites, say 1 or 2 parasites in a billion RBC, you might not spot them by microscopy as well as by your PCR. But those parasites, once you infect to a naive non-immunized mice, they should pick up and they can infect. And then you will know that those mice are really not negative, but they are positive. Okay. So what we did was we transferred the blood from this immunized and protected mice to naive mice and followed the parasitemia for 8 days. And we found that exactly the same replication, the data that we had, the 2 mice that were positive here and they came up here, they were they just transferred to naive, but all the naive, all the protected mice, they didn't. So in case of ATG8 immunized, none of the mice get infected. So they are really protected. So this like intriguing like results like prompted us to study further what is happening, because why it is like better. And this is another study that with a long term, like 6 months, we saw again the similar pattern, that we saw the ATG8 CVEC immunized mouse are protected better. More mice are protected as compared to the wild type. So far, the wild type CVEC approach was the best. So we found that when we combine that with a mutant that stays longer in the liver, it is even better. So we designed another experiment where we tried to check, because sometimes what happens is the protection is best when you are challenging with a homologous strain, like the immunization strain and the challenge strain. If they're same, then it's fine. Like you get infected, you are immunized against falciparum, you got a challenge with falciparum, you might see better protection. But you immunized against falciparum, but you got challenged with vivax, you might not get protected. Okay, so to test that hypothesis in the rodent parasite, what we did was we immunized them with 3 doses of this parasite, and then challenged them with a different strain, different species of parasite called Plasmodium eolei. We used Plasmodium berghii for immunization, but challenged them with Plasmodium eolei during the memory time point. And we saw a like better protection as compared to the wild type. So it is not only giving better protection against the homologous parasite, against heterologous parasite as well. And then we went on to study what are the immune response that are responsible for the protection. And for this, what we did was we immunized them for 2 doses the way we did earlier, and then we deplete the immune cells, different immune cells. Basically, we deplete the CD4 T cells and CD8 T cells by anti-CD4 or anti-CD8 antibody. And then we looked at, once we challenge them, these mice after depletion, if they lose protection. So that's how you will know that which cell is important for their protection. So in this graph, it is very like complex graph. So you can see that the infection control, all of them came positive. And this is the wild type CVEC, ATG8 CVEC. These are basically immunized and challenged normally. Okay, they all are protected as I showed earlier. And then what I did was, this is the two group in orange and in purple, where we mock deplete. We use a control antibody to they will not deplete any CD4 or CD8 T cells. And this group in purple, it deplete all the CD4 T cells, and the one in blue, all the CD8 T cells. So once you deplete these CD4 and CD8 T cells, you see the mouse start losing protection, and they lose, most of the mice they lose protection when you deplete CD8 T cells. It tells us the CD8 T cells are important for the protection when we immunize them with the ATG8 mutant. So now we know that CD8 T cells are important for protection. So how they are doing this, their effector function, how they are protecting the CD8 T cells. So to study that, one of the things that normally in malaria field that we know that is CD8 T cells kills these infected hepatocytes by secreting inflammatory cytokines called interferon gamma. Okay, so whether interferon gamma is playing an important role here or not, to test that hypothesis, what we did was we used normal mice, and this group on the left panel you are seeing is uh, normal mice. They are not immunized and but challenged with a wild type parasite that express a protein called luciferase. And when you inject the substrate, you can see the light coming, and you can, without killing the mice, you can estimate the burden of the parasites. So they are all infected in the liver. And in this imaging that we do after challenging the sporozoites, 40 hours after challenging, we image them. So we can see them, they are developing in the liver. So what we did here in the wild type mice is we immunize them. So they are all protected. Okay. So this is the wild type mice. And then we used interferon gamma knockout mice and interferon gamma receptor knockout mice. And we see in the interferon gamma knockout mice, we see there are little parasitemia in two mice, and the level of parasitemia is low. I will show you in the next graph. And interferon gamma, none of the mice got infected. So it tells us that interferon gamma is not important. It is just behaving like this, like the wild type mice immunized. So CD8 T cells is important, but they are not killing the parasite infected cells by using interferon gamma. That's the message that we are getting from here. And this is what I'm showing here. This is basically the parasite burden, as you can see in the infection control. This is just the graphical representation of what I was seeing on the left side. And this is what you are seeing by blood smear, whether they're positive or negative. Here you can see that parasites they're coming. These two mice, they're positive here because they got infected. So we can say that interferon gamma is not playing a significant or major role in this. So there might be other components like porphyrin and granzyme. These are other effector molecules that use by CD8 T cells to kill infected cells. But in our approach so far, we see this is different than what people have seen. So that's why it is happening. We need to study it further to design a better vaccine. Then what you are seeing here is we immunized, we studied the CD8 T cells, CD4 T cells, and CD8 T cell is important. Now we wanted to see how the antibody response, whether we are developing any antibody response against what kind of different stages. So what you are seeing here is this three line represents the immune serum from naive mice, those non-immunized, wild type CVEC mice, and the ATG8 CVEC mice, our mutant. And these are different parasites. This is the blood stage, the leftmost panel, and this is the sporozoid stage, that is the form that transfer to human being or the mammalian host from the mosquito. And this is the liver stage at different time point because we are targeting the liver stage. So we saw that what you are seeing in the green staining is basically the immune serum that is reacting to the parasite antigen. So you can see naive, you don't see any reaction to any stages. But wild type CVAC and ATG8 CVAC, basically you see all the stages are being detected by the antibody. So this antibody are detecting all these parasite stages. So the next question is whether this antibody has any functional significance. They can kill this parasite, they can protect us, protect the host from infection. To test this hypothesis, what we did was we designed an experiment where you take this sporozoite, the form that infect the host, we incubate them with the immune serum, and then we inject them to the naive mice and see if they are protected. And then I follow them for the parasitemia because if they are successfully infecting, they will lead to the bloodstream infection. And what we observed was these two, your black and blue in this graph, are the infection control and naive. All of them they came positive. But with this wild type and ATG8 CVAC, none of them became blast stage positive. This tells us that these antibody are protective. They can bind to the sporozoid and neutralize them. So they cannot cause successful infection, blast stage infection. So now we know that antibody are also protective. So to summarize whatever we have seen so far is that we saw the ATG8 overexpressor is it stays longer in the liver and that's developed poorly, and also it induces a strong memory response as opposed to the wild type. And then it because it strong induces a strong memory response, it confers long-lasting protection. And this protection is CD8 T cell dependent, and CD8 T cell mediated protection. This CD8 T cell does not use interferon gamma for the protection, and this also our ATG8 is also able to provide cross species protection, which will be a better vaccine if we design clinically and test it in humans, because that's what you want. So that if you immunize with one vaccine, it should protect against all different species that infect human being. Currently, there are five different species they are infecting human being, like Plasmodium falciparum, Plasmodium vivax, Plasmodium malariae, Plasmodium knowlesii, and Plasmodium cyanomolgi. All these five species are infecting human being. So if we immunize against one, if they can protect against five, then we are better protected. So we need a vaccine like that. So we have to use an approach that is going to give us this kind of protection. And this ATG8 CVAC, this induce a humoral response, the antibody response that can detect antigen against all different parasitic stages, and also they protect against infection. And with that, I would like to thank people from our lab, my supervisor Isabel Coppens, and all the lab members, past and present lab members, and the like Sinis lab from our department. They are expert in the sporozoite uh, field, and they helped us a lot in designing some of the experiments. And the Javala lab, Fidel Javala, he's an immunologist, and their lab gives us guidance on how to do all this immunology on mouse models. And none of the experiment would have been possible without the help of the Insecti Core facility. And Godfrey, Abhay, and Chris, they were like rearing the mosquito and providing out infected mosquito. And most of the study that we did was supported by funding from National Institute of Health and JHMRI, this Johns Hopkins Malaria Institute pilot grant. And thank you for your attention, and I'm happy to take any questions.
Thank you, Dr. Tejram. And it is very informative. And we got in-depth knowledge about the vaccination strategy of the malaria. And I would like the participants to write their questions so that we can ask and we'll get the answers from the Dr. Sahu. Who has questions, they can raise their hands and they can ask on the chat section.
And I'll stop sharing, right?
I think if it is needed,
Okay. Okay.
is there any questions from the audience? Okay, Abhishekta.
Good
Abhishekta.
morning, sir.
Yes.
Actually, I have a doubt. In this experiment, instead of the infected mosquitoes, cannot we use the infected malaria patient's blood or cell line something?
Uh, Actually, I hear part of the question. So, instead of infected mosquito, what you wanted to use?
The malaria patient's direct blood cell line.
Yeah. So that's the tricky part because that's the reason that I touched upon the bottleneck of the parasite. Let me go back to that one actually. The bottleneck, the strategy that I'm proposing is against the liver stage, because as you can see here in the blood stage, the number of parasite is huge and the parasite life cycle is very short, like 24-hour cycle. And in 24 hours, the parasite is producing like millions and millions of parasites. So it will be hard for controlling them. And there are people have used this approach against the like they use the similar approach like the chemoprophylaxis, but against the blood stage. It is working. It works, but the impending uh, threat for that approach is that now the malaria parasite is developing resistance against most of the drug, including the artemisinin that is being used as a frontline drug. So when you are using that, uh, it is hard to develop a kind of drug that's going to kill them completely. So if they cannot be killed, this vaccination approach will fail. So that's why it is not being considered as a viable approach.
Okay,
Thank you,
Ankita,
sir.
Okay.
your
Good
question.
morning, sir.
Yeah.
Sir, belonging from Odisha, I want to ask that how to increase awareness and scale up malaria elimination program activity, especially in Odisha.
Yeah, that's a very good question, and I have thought a lot about it. Uh, and I think it will be better if we include uh, this kind of awareness program in the school level, and like invite like people, do like frequent meetings, like hold presentations and seminars in at the school level, and make children aware about this the threat and the problem. Then definitely they can contribute a significant way by managing their environment. It's a problem of like cleanliness. Like people have water, like stagnant water for in a lot of places. Okay, they just ignore, and that's the perfect breeding ground for the mosquito. And once the mosquito, they get the parasite from infected humans. So if you eliminate that, then you control significantly. And in addition to that, like it will be better uh, to invest in uh, in research and development. But that will definitely depend on the government. So we have to work in policy level with the government as well to implement those policies. They'll fund more towards developing in vaccine research, infection vaccine research. These are the two things that I would think that should be done. And particularly Odisha, Nabin Patnaik government is aiming to eliminate malaria in Odisha by 2030. So that's a kind of very highly optimistic goal, but I don't know how it can be achieved at this moment.
Thank
Say,
you, sir.
what is your question?
Morning, sir. Am I audible?
Yes, yes, please.
Sir, I actually wanted to ask you two must have heard about the CRISPR technology. And in the sub-Saharan African region, they use this technology to actually alter the genome of heroes when the spread of malaria. So it can also be done in India.
Can you like I couldn't hear because your voice is so
Yes.
low.
Could you please write it if it is, or you can increase the voice, the volume of your speaker.
Sir, am I audible now clearly?
Yes,
My loud
please.
enough?
Yes.
Okay, sir. So you two must have heard about the CRISPR technology. It was
Yeah.
Okay.
So in the sub-Saharan African region, they introduced this technology in the gene, like they altered the genome to prevent the spread of malaria. Like they caused the mutation in its genome and made its bite ineffective. So can this also be done in India? That we will not be in need of vaccines.
Yeah. Yeah. So it's a very good question, and I didn't touch upon that actually. Honestly, I didn't touch upon that because of the time. So now there are like a diverse array of approach are being employed. Particularly in sub-Saharan Africa, they are using this CRISPR technology to develop gene drive. So the gene drive is a kind of technology where normally what you do is you introduce particular gene that's going to favor either only male or female progeny. So they like male sterility, or they will favor like cue the progeny into either towards male, or they will make all the males sterile. Okay, so this is what they do. So
Yeah. Disconnected. So someone, it is possible that someone disconnected.
hello.
Yes, you are audible.
Yeah, yeah, I have been removed, so I don't know how I've been removed. Okay.
Yes, I think this is someone by mistake has done it. Okay, continue.
Okay, okay. So that's called gene drive technology, where they modify the genome uh, of, of the mosquito, then they introduce them in the field. They will compete with the wild type male, and then the next progeny, successive progeny, will be like sterile or male skewed, so that they will not transmit the disease. Yeah, it is possible. In Africa, there are like three different kind of species, like Culex, Stephensii, and Gambi are dominant. But in India, there are like several species. There are like, I think, more than 150 different species. They are responsible for the transmission of the disease. So you have to, it is doable, but you have to develop that diverse uh, gene drive on diverse number of species. So it will be possible, but you know, transgenic living transgenic mosquito without proper like long-term study, it is kind of very hard. So we don't know what is the long-term effect of this, and also like how successful this gene drive is unless and until it is properly validated. Sometimes it works in the lab environment perfectly fine, but when you leave them into the environment, because the lab environment is like you control everything, like your temperature, food, your microbe, and everything. And the moment it goes into the wild, all these variables can affect the outcome, and it might be counterproductive. So though it is being rolled out some part of the Canada and some part of the Africa, it is in the kind of infancy of this approach. But definitely it can be doable with time, and it's getting better and better with time. So one day it can be used in India as well.
Okay, thank you, Dr. Sahu, and thank you, Sheikh, for this very important question. Next question is from the Devi Prasad. Devi Prasad, your question, please. Devi Prasad, is your internet okay? Your question, please.
Yes, sir. Could you please box lyrical vis-a-vis whether our physic could dial down malaria sans any medication?
Can you repeat it, Devi Prasad?
Sir, could you please walk lyrical vis-a-vis whether the physic, our physic could dial down malaria sans any medication?
Our physique, uh,
Can our body cure against malaria without any medication?
see, uh, I don't think at the moment you can like cure malaria without any medication. So that only you can do when you are vaccinated, so that your immune system will eliminate the malaria. So with if you are not vaccinated and you are susceptible to malaria infection, then I don't think that just with your physique you can eliminate malaria. No.
Abhishek, your question? Do you
Sir,
have,
what is the difference between malaria and brain malaria?
Yeah. So yeah, malaria and brain malaria. Malaria definitely is caused by the parasites, and brain malaria is also caused by the parasites. But the brain malaria happens when the parasites cross the blood-brain barrier and start causing inflammation into the brain. So that normally happens sometimes. Like it depends on the species and also the immune system of the host, how reactive the immune system is. So depending on that, the Plasmodium falciparum can cause brain malaria, which is called cerebral malaria. So it's basically the same malaria, but the parasite sometimes it can cross the blood-brain barrier and it cause inflammation in the brain. That's when it's called brain malaria.
Thank you.
Okay. Thank you, Dr. Sahu. And can you please tell us that when the common people can use the vaccine, malaria vaccine, like us?
So see, I think just last month or maybe a month and a half before, the World Health Organization has licensed like uh, uh, for this RTSS vaccine to be given to all malaria endemic people, malaria endemic country. So right, it's so far it has been like used or people were getting from Africa, they were getting the malaria vaccine, but now on probably everywhere in the world, wherever the malaria is endemic, they can get this vaccine from GSK. Yeah. So, but that
And
vaccine is only 33% effective, and and looking at in India how effective it will be very hard to say because in India there is no malaria immunity against there is no immunity against malaria even in the adult. So it will be hard to say, but definitely the people will get in India this vaccine.
in this vaccine dosage, but is there any booster dose? We need to take one dose or multiple dose like this?
I normally what they are giving at the moment is I think it is 3 dose malaria
Hmm.
vaccine. Okay,
Hmm.
but it all depends on the transmission level and how effective it is in that setting. So far they haven't tested in India, so they have to do testing in India, then how effective, then based on that they will recommend the dose.
Okay, so till now the malaria vaccine effectiveness is 33%, right?
Yeah, the RTSs. Recently
Okay.
there is another one in phase 2 clinical trial. There's another group in Oxford. They developed a vaccine called R21 M matrix. So they used a different protein than the protein they used for RTSs, and it is 77% effective. And so if that vaccine works well, then definitely it is a kind of very positive sign. And but it is
Yeah,
in phase 2, so it has to go through phase 3 trial, then a lot of regulations. After that it will be approved.
at least two
So
to
once
three
it get
years.
approved, yeah.
Devras, sir, do you have any question?
Okay, thank you, sir. I have some question. Thank you very much, Mr. Pavu, for giving a nice informative talk. I have some query. What I would tell you that
Yeah,
Plasmodium vivax,
yeah,
general malaria, has been caused by Plasmodium vivax only, and the brain malaria is caused by Plasmodium falciparum.
yeah,
General,
that's what I'm saying. Yeah, but Plasmodium falciparum not all the time causes brain cerebral malaria, not all the time, but mostly Plasmodium falciparum causes cerebral malaria because it can cross the blood-brain barrier. It also depends on the host status, host immune status. But Plasmodium vivax doesn't cause brain malaria, that's for sure.
Okay, sir. Thank you. That's
Yeah.
why I have because thank you very much. How much? What is the cost actually? Cost or this is giving free to the people of Africa?
So, so far, because they were testing it in Africa among the children, it was free. But once it will come as a vaccine, I don't know what will be the cost. That the government and the company, because the company has already spent $3 billion on this vaccine, so definitely it is not going to be free.
Okay, thank you. Thank you.
So if it's a commercial company, right? They're investing money, so they will get their money back because they're putting money. So definitely somebody is going to buy, whether it is World Health Organization or it is government. Somebody is going to buy, but they might give you free to people. But like we might feel it is free, like right now COVID vaccine. We think that it's free, right? We are getting it free, but somebody is buying, right? The government is paying the money to to this company, Pfizer or BioNTech or Moderna. They're paying them. They're buying it. They're giving free to the public, but they are taking money in terms of tax. So it is a kind of you don't feel it, but they will take it from you.
Oh, okay, thank you very much. So nice. Thank you very
We,
much. But I would like to invite you in another occasion in
Sure,
our lesson. Thank you.
sure, no problem.
will if he doesn't mind, we'll keep bothering himself for many lectures series.
Thank you.
Okay,
Thank you.
sure,
Thank
sure.
you.
Shweta, Shweta, do you have any question? You had raised your hands, now your hands is lower. Shweta,
Yes, sir. Sir, actually, my
okay,
question
what?
is there a medication things that anyone can prevent themselves before vaccination?
Yeah, so there is a preventive medication that normally a lot of the people from here, because in I am working in US, but people in US, they don't get malaria. Okay, but they, when they go to like malaria endemic country, they take prophylaxis, drug prophylaxis beforehand, and once they reach there, in their system the drug is there, so it will prevent them from getting infected. Okay, so that's normally people use the chloroquine and this artemisinin, and there are like other like there's a combination therapy like artemisinin and amodiaquine. So they combine these drugs. So uh, they take it in advance, so that this is called preventive prophylaxis. But that, whether it can be done in India, it is hard. I don't think so. The reason I don't think is because malaria is not seasonal in India, and the giving drug beforehand has its own side effect. The problem with the drug resistance is because people were using this drug like improperly. So like either they are taking this drug when it is not required, or they are not taking it sufficiently, or they're taking it partially. So that's when it is more dangerous. The parasite will have slight exposure of the drug. So we do similar kind of experiment in the lab. We treat them suboptimal concentration of drug frequently, and then after several cycle of the replication, they start developing resistance against the drug. The same thing happens in the field. They treat people, and they treat people with the drug, and they don't know that whether they will get exposed and how much drug will be there in the system. Say you took the drug, you given to the you gave people to the drug, and then say the drugs lasted in their system for say 5 days or 10 days, but they got infected on day 9. Okay, and then what will happen? Like they will have a little drug in their system, so the parasite will get exposed to this not lethal concentration but sublethal concentration, and this it happens repeatedly. The parasite will develop resistance, and then it will be kind of dangerous. So this is not going to work in India because it is not seasonal, but it might work in seasonal like in Africa, where before just the rainy season you can give them the drug, and in rainy season, throughout the rainy season, you provide them with the drug, so that they will maintain a high concentration of this drug in their system, so that they will not get infected. So it will work there, but not in Indian setting. It's not going to work.
Sarada,
Good morning, sir.
Yeah.
My question is, does malaria affects animal?
Yeah, yeah. Malaria affects animals because see, we study rodent malaria. Rodent malaria is basically the malaria parasite that affects mice or rats. So there are different like malaria against all the different kind of animals, against birds, cattle. So they also get malaria, but like because they can't tell us, so we don't know that they have malaria, right? That's the thing. But there are
Prad,
malaria species, different species. They affect infect different kind of mammalian host. Yeah.
Pradumna,
Good morning, sir. Sir, I have a question that between malaria and dengue, which is more dangerous?
Between malaria and dengue,
Yes, sir.
actually both are dangerous. But dengue, you can see that like people get very quickly sick, and because their platelet get like depleted, people die very quickly. But with malaria also, there are people are dying very quickly, depending on the severity and how which parasite is infecting them. So with falciparum, a lot of people they die. A lot of children die with falciparum infection. So comparison is it's kind of relative term. It all depends on how robust your immune system is. So it's there is no direct comparison between these two. I cannot say like there will be a direct comparison. But dengue is definitely, but both are transmitted by mosquito. One is your viral disease and one is your parasitic disease. So both are deadly.
Sir, another question, sir. Is the brain malaria is dangerous than the normal malaria?
Definitely brain malaria, because a lot of people they go run into coma and they die because of the inflammation in the brain. So brain malaria is more dangerous for sure.
Sir, why?
Bhakti.
What the main mechanism about it?
So the parasite somehow like it breaches. Normally we have in our brain there is a kind of our brain keeps everything separate from rest of the body through a membrane. Okay, and not all the things can pass through the to the brain. Uh, Definitely not all the immune cells can go to the brain, because it is immune privileged part. This called immune privilege. There is a concept that probably it's an immunology concept. I'm not going to describe why it is called immunoprivileged. And it normally avoid all the inflammatory processes. That's what I can say in a kind of very simplistic way. So somehow the parasite can breach that barrier and can go inside. Once the parasite goes there, then all your immune cells, these inflammatory cells, can go into there, and they try to start attacking them. And that's when it created the inflammatory environment in the brain, and then the brain get damaged. And as you know, the most of the brain cells, they can't recover themselves because these are neurons, long-term neurons, and if you get damaged, there's a permanent damage. That's why it is more deadly.
Thank you,
Bhakti.
sir.
Yes, sir. Good morning, everybody. Sir, my
Sir,
question is,
Pranam.
is the proportion of vaccine same for children as well as adults, or it is different?
The proportion actually for children, for adult it will be different, because the status of the immune system is different in children and the adult has a more developed immune system, and the children, they are developing immune system. So the dose that are being used are different, and the adjuvant that you can use will be different, and the duration between the immunization and booster dose will be different, because it if you use the same as adult in the children, then it might be cause severe reaction. So it will be different. Yeah, the one line answer will be the different.
Pralay.
Good morning,
So,
sir.
sir, at what age the
Okay,
children can be given
Bhakti.
this vaccine?
So right now they are rolling out this vaccine 5 month plus. So once the children are 5 month positive, like they cross the 5 month, they can receive the vaccine, because that's when most of their immune system are developed.
And sir,
Yeah.
you said as it also can affect the animals. So, is it different for animals even?
So for animals, we don't develop any veterinary vaccine yet, but definitely right now, most of the research community is focused on developing a vaccine against human malaria. That's the priority. But we use animals malaria as a model to understand, because see, a lot of the things that you cannot study in humans, right? So blood stage you can study in the culture, but the liver stage, it's very hard to study, and for that you need primary hepatocytes. So for this kind of things that we use animal model, so we understand animal parasite better than human parasite as because you use them to study each and every part of them. Yeah.
Is it
Okay, sir.
enough,
Thank you so
Bhakti?
much.
Pralay.
Sir, sir, why malaria parasite does not infect mosquito?
No, they infect mosquito. Definitely, they infect mosquito, because in the mosquito they reproduce. Their sexual cycle happens in the mosquito. And as far as like your question is, I think whether they die because of the infection,
Yes.
right?
Yes.
Yeah, they're like significant. Their life longevity get reduced because of the infection. The problem why they died, not because of the malaria parasite cause any uh, damage. The only problem because they die because they cannot feed. What happens is the malaria parasite See, the way of transmission is through their proboscis, right? They get blood, they get their food through the proboscis. Okay, so what the malaria parasite does, it clogs that proboscis, so they cannot get their food properly, all because it is choked with the parasite. So that's why they need to feed multiple times. So because of the
Okay,
hunger, they die, not because of the parasite.
sir,
Copy Sreda.
why the
Yes.
vaccine took a longer time to be prepared as there was high mortality and morbidity rate at that time?
Say that again.
Sir, why vaccine took a longer time to be prepared as there was high mortality and morbidity rate at that time?
Yeah, so I think if you saw that the problem that is associated with developing a vaccine that I described as virus, the bacteria, and the parasites, the complexity of the parasite, because malaria parasite is an eukaryote and it has different shape, shifting, and as it shifts from one form to another, it changes its expression pattern, like it express a different set of protein. So which protein you are going to take as a vaccine candidate? That's the first question. The other question is, like, what if you change, if you selected a protein and the parasite, because parasite have multiple forms of this protein, then what it will do is it will replace that protein with another one. Okay, so the best example for that is there's another parasite called trypanosoma. So what it does is it will coat with a called protein called VSG, various surface glycoprotein. It will coat with itself. So once the host mount immune response, it will change to another VSG. Only one parasite will change to another VSG. So that will escape, and all the other parasites will get killed, and that one parasite then replicate, and it will evade the immune system. The similar things happens with the plasmodium because it coats itself with the form that goes from mosquito to human. It coats itself with a protein called CSP. That is the protein that is now being used for the vaccine, because it is not effective, because the parasite when it moves it sheds that protein. It normally it just like removes like a cloth. So imagine the antibody is bound to it and then it removes the cloth. So this antibody is of no use. The parasite can easily go into the cells and develop. So, and it has to do, only one parasite has to do that thing successfully. And once inside the cell, your antibody is not going to go there and kill the parasite. So it is basically, you have to target a needle in the haystack, and that too, the paras it is changing its cloth all the time. So you have to design a vaccine that's going to target it in quickly and effectively. So that's the complexity. Because of this complexity, it is very difficult to develop a vaccine. People have been working on this field for almost 60 years. And now we have a vaccine that is 33% and some are 70% effective. And we know there is a vaccine that is 100% effective and lasts more than 2 year protection. But logistically, it is not possible to use that as a vaccine. And because the antigen that we are using, you have to grow billions of mosquito and then dissect them manually and take the sporozoid, because you can't culture them in culture. That's the problem. Like virus and bacteria, you can put them in a culture and you can grow them. But the vaccine that we are talking about, you cannot culture them in the culture media. That's the logistical problem. Once we are able to do that, people are working on that definitely, but I don't know how long it will take. But eventually, probably we'll get there.
Okay,
Okay.
sir.
Copy Sreda. This should be the last question. Copy Sreda. Are you there or not? Okay. So thank you, Dr. Sahu, for short notice and giving such a beautiful talk and making us enlighten about the malaria vaccine and attending all the questions of the students. Now I would request Dr. Masumi Misra to present a vote of thanks. Please, madam.
Good morning, one and all present here. Am I audible properly?
Yeah.
Okay, sir. I want to express my sincere appreciation to Professor Arvind Desmukh, sir, for taking out time from his busy schedule to grace us with such encouraging and appreciating words. Thank you so much, sir, for your thought provoking and valuable address. And I would like to thank our distinguished speaker, Dr. Tejram Sahu, for making an excellent presentation and making the seminar a very meaningful, valuable, and interesting one. If I heard right, sir, 77% efficacy of malaria vaccine is a very encouraging condition, sir. We
Yeah,
hope it will be successfully designed soon. And if I'm not wrong, it will be the first parasitic vaccine to be designed, sir.
yeah.
Okay, sir.
First,
Then,
eukaryotes. Yeah.
okay, sir. I would like to express our deep gratitude to Professor Niranjan Behra, sir, for his valuable suggestions. He is unable to attend the meeting though. And I would like to thank all the coordinators, Dr. Dheeraj Parida, Dr. Debraj Parida, Dr. Dheeraj Nanda, and Dr. Sushil Kumar Sahu for taking out time and participating in the seminar. And I would like to thank our director, sir, Professor G.K. Panda, for his guidance and moral support and motivation. Though he is a very busy man, he always take out time for active participation. Thank you so much, sir. I would like to thank Dr. Satyanand Patel, coordinator for MSI for MSP, and for taking all the pain to make this seminar happen. And at the end, I'm happy to express a vote of thanks to all our staff and students for their queries and interest that has made this seminar a successful one. Thank you all again.
Thank you.
Thank you, Dr. Sahu.
Thank you. You're welcome.
So along with it, thank you, Dr. Sahu again. Thank you, Director Sir, and thank you all the participants. We can close it now.
Thank you. Good night. Good night according to my time actually, and good morning.
Yeah. Good night from my side. Thank you, and we may bother you again for such type of lecture.
Sure. Okay. Bye.
Bye.